Next, we will talk about characteristic of the solution, such as pH of the solution, how it's called. So pH solution is a concentration of the hydrogen ions in the solution or amount of these ions. So if solution is have pH 7, it's called neutral, and it means it have same amount of the hydroxyl and hydrogen ions. Amounts of the hydrogen ions same as amount of the hydroxyl ions. So, and what is this will be? This is will be pure water example of this. The other solution we can say black coffee, acidic rain, soda, hydrochloric acid, gastric acid, battery acid. So in this case, we can see that pH will be less than 7. If it's less than 7 pH, it means this solution is acidic, and it means that amount or concentration of the hydrogen ions is more compared to the concentration of the hydroxyl ions. So this is acidic. pH will be lower than 7. But if solution will have pH more than 7, so in this case, this solution will be alkaline or basic solution, and it means it will have more hydroxyl ions compared to the hydrogen ions. An example of this: drain cleaner, bleach, soapy water, ammonia, milk of magnesia, baking soda, and seawater. So this is what, and usually in the lab, we have this is paper, and if you put this paper deep in the solution, some kind of, it can change color. So if color, for example, will be like this, you will say, oh, this is pH 7, because this is will be on the this paper lacmus. It will change color when you dip in different solutions, and it's have color coded in here what is the pH, so you can match and say immediately what pH of this solution. But if you dip and it will be red, you say, oh, it's pH 2, for example, if it's like this color. Or if it's like this purple, violet, so you will say, oh, pH 11. So this is how it's work usually. So now you need to define what is a pH. Next, we will talk about different solutions, type of the solution. So it could be isotonic, hypertonic, or hypertonic. Iso means the same, tonic. Hypo, low, tonic. Hypertonic, high, tonic. So usually when we're talking about this, we're talking about concentration of the salt in the solution compared to the cell. Yes, and in here show different cells. This is cell 1, cell 2, and cell 3. So if we will put one cell, this is cell number 1, we put it in the some water solution, but it's have a little bit of the salt, and concentration of the salt will be less than 0.9. So in this case, this solution which surrounds the cells will call hypertonic. Hypo means low tonic. So inside of the cells, usually we have concentration of the salt 0.9%. And this solution in here, we put cell 1 inside 0.9% of the sodium chloride concentration of the salt inside of the cell. But we put in the solution which is less than 0.9. So in this case, water from the solution in here, outside part, will move inside of the cell. And cell will increase size and it will be continuous until it will burst. In here we have holes and water completely coming inside of the cell. So this is what happening during osmosis. So osmosis is movements of the water. Osmosis. And water always moving from the area of the low concentration of the salt to the area of the higher concentration of the salt. So if we put cell in this solution, hypertonic, low tonic, its mean concentration less than 0.9. For example, it will be 0.1% of the salt. And inside of the cell will be 0.9% of the salt. So we have less salt in here than inside of the cell. Outside of the cell 0.1, inside 0.9. So water always moving to the higher concentration. How they say water follows the salt. So water will move inside and cell will increase size until it will be damaged and broken. Yes. So increase input of the water in the cell cause lysis of this breaking down of the cell. If we will put cell in here, cell number 2, inside of the different solution, it's called isotonic solution. Iso means the same. So it means concentration of the salt inside of the cell will be the same as a concentration of the salt outside of the cell. So this means this solution is isotonic. It's same as concentration of the salt inside of the cell. In this case, water will move in, water move out, but it will not damage the cell. Cell will not increase size in here and will not go through lysis and damaged. So this is what's happening with cell in the isotonic solution. And if we put cell number 3 in the hypertonic solution, hyper means high concentration of the salt. So it means inside of the cell we have always 0.9% of the salt. And this solution around it, hypertonic, so it will be more than 0.9. So it could be, for example, concentration 5% of the salt inside of this solution. So in this case, water will follow the salt. Water will move from area low concentration of the salt to the area of the high concentration of the salt, away from the cell. And what happening with size of the cell? It will shrink. Cell will lose water, it will shrink, and it will be damaged because of that. So now we will talk about, so when you talking about this osmosis in different solutions, isotonic, hypertonic, what one important rule you need to remember? Water always follows the salt. Water will always move where is higher concentration of the salt. Okay, so plasmosis is a process when high osmotic pressure It's happening in the hypertonic solution. Remove water from the cell and cell shrink. And it's happening where? In here. And we put cells in the hypertonic solution. So happening plasmosis. High osmotic pressure or tendency of the solution to uptake water inhibit growth of the bacteria. And this is what is used, this osmotic preservation of the food. For example, when we're making fish salty to preserve it, salty surrounding means that bacteria cannot survive in here. They will go through the plasmosis and die. So this is one way to preserve our food. Honey, sweetened condensed milk, jam, all this, the other example, it's not only about salt. High concentration of the sugar in the honey, in the sweetener, in the jam also will make the same. And in this case, water will follow the sugar, whereas more sugar and cells again will go through the plasmosis, shrink, and bacteria will die. So this is how preservation of the food happening. So now you need to think about this. Will size of the piece of the potato change when you put it in the isotonic, hypertonic, or hypertonic solution for several hours? So right now think that you cut very thin slice, very small and thin size of the potato. And potato is a living organism, so it's made up of the cells. And you put it in different solutions. Yes, you put your potato inside of the solutions, different solution in the water. We have 3 different solutions in here. So one solution will be isotonic, the other hypertonic, and third hypertonic solution. And you put like this little piece of the potato for several hours. So now you need to think what will happen with size of the potato. These cells, these pieces of the potato made up of the cells. So if cells change size, potato, this little piece will change size also. So think about this, draw picture, and we will discuss this during our lab.